Good morning, good afternoon, good evening. Thank you for joining us for today's ISAC webinar. I'm Jinxin Zhao from University in Melbourne, Australia, and it's my pleasure to welcome you all to this session. Before we start, I would like to briefly introduce the ISAC and its early career working group. ISAC, the International Society of Antimicrobial Chemotherapy, is a leading global organization dedicated to advancing education and scientific research in infection management and antimicrobial therapy. With over 90 affiliated member society and more than 50,000 individual members worldwide, ISAC plays a key role in connecting professionals in the field. If you would like to know more or get involved, please feel free to reach out. ISAC includes 21 specialized working groups. Our early career working group is committed to building a platform where ECRs can connect with senior experts, exchange ideas, and develop collaborations across the globe. We are passionate about creating opportunities that foster communication and long-lasting partnership within the community. Our leadership team actively works alongside ISAC and early careers to design programs and organize events that support professional development. If you are interested in contributing ideas or becoming involved in future activities, we warmly invite you to join us. Now it's my great pleasure to introduce our distinguished speaker, Professor Hinrich Schulenburg. Professor Schulenburg is a renowned evolutionary biologist, a fellow at Max Planck Institute. His pioneering research in experimental evolution, genomics, and mathematical modeling is widely recognized, and he has been instrumental in applying evolutionary principles to tackle real-world challenges, particularly in antimicrobial resistant. Please join me in giving a welcome to Professor Schulenburg, who will now lead us into today's talk.
So it's a great pleasure for me to talk to you today. And I will talk how we can use evolutionary concepts to optimize antibiotic therapy, and with a particular focus on sequential antimicrobial therapy. I will briefly start by introducing myself. So I'm an evolutionary biologist and working at Kiel University and the Max Planck Institute for Evolutionary Biology in Plön in northern Germany. My group is working in general on the evolution of hosts and microbes. We do a lot of work using C. elegans as a model host, where we look at C. elegans microbiota interactions or C. elegans pathogen coevolution. And part of my group is working on antibiotic resistance evolution, and that is the topic of today. I'd like to emphasize, since most of you may not know where Kiel University is or Plön is, it is in the most northern state of Germany, directly on the coast of the Baltic Sea, and has become one of the hotspots for evolutionary research in Germany. And this is thanks to a couple of larger research consortia and also including a new research building, the Center for Translation Evolutionary Biology, that is currently being built and will be ready for us to move in 2027. And I wanted to highlight that we will soon advertise a new junior group position that is focused on the translation of evolutionary principles into antimicrobial treatment strategies, more or less the topic of my talk today. So if you're interested in this, send me an email or keep an eye out for an advert that we hope to publish in the middle of the year. So if we think about evolution-informed therapies, and especially evolution-informed sequential therapies, then there are a couple of concepts that are exploited. On the one hand, fast changes in the application of antibiotics make adaptation of pathogens challenging and difficult. And that is illustrated with the graph here on the left. So fast changes really means that we change between antibiotics every 24 hours or faster. And this then means that the selective pressure imposed on the bacteria changes rapidly. And we know from evolutionary theory that this makes it difficult for organisms to adapt in general. On top of that, we may be able to exploit evolutionary trade-offs. And I will introduce one of these trade-offs to you a little bit later in my talk, a trade-off that has been termed collateral sensitivity, and that shows particular promise in improving antimicrobial therapy. And another phenomenon is exploiting, again, physiological changes at antibiotic changes. And this has been named negative hysteresis. And again, it's a concept that I will introduce to you in a minute. But this is the overview of current concepts that are currently considered to improve evolution-informed therapy. What is important for the application of such novel concepts is that they are broadly applicable so that we can actually apply them and use them without having too much information on the patient or on the pathogen, because usually we need to implement treatments fast when a patient comes into medical care. So this is something that we try to evaluate. My group is a basic research group, so we do our work in the laboratory, and we especially use evolution experiments to compare alternative treatment regimens. And we use experiments, these kinds of experiments, to also be able to infer cause-effect relationships. We combine the evolution experiments with genomic and genetic analyses to have a better understanding of the exact targets of selection, of antibiotic selection. And at the moment, we do most of our work with Pseudomonas aeruginosa, one of the most important critical human pathogens. And of course, here the aim is that our laboratory work will then have some link to the clinic and may facilitate any kind of clinical intervention in the future. So sequential therapy is actually not used that much, even though in our opinion it shows particular promise. And this is the reason why we try to investigate resistance evolution under antibiotic changes under sequential therapy in detail. And the first result that I'm showing you today is based on experiments that were performed by a former PhD student and then postdoc, Roderich Röhmheld. And here we had a very comprehensive evolution experiment where we changed between 3 antibiotics from 3 different classes: carbapenem, gentamicin, and ciprofloxacin. And this is the general setup of the experiment. Horizontally, you see sort of the time of the experiment. In total, we had 96 transfers, and this amounts to approximately 500 generations of the bacteria. And then vertically, you see 4 main treatment types. On the top, monotherapies as a control, so no changes between antibiotics, only application of 1 antibiotic. Then you see fast and slow regular changes between antibiotics. And at the bottom, you see a random change between antibiotics. These were the treatments that we then used. And then we assessed to what extent bacteria are able to adapt. One readout that we used is extinction of the bacterial populations. Here it is important to emphasize that we always applied antibiotics at sublethal concentrations. So this should usually lead to survival of all the evolving populations. But interestingly, we did observe extinction of populations. So this is a sign that the selective conditions of the treatments are too challenging for the bacteria to be able to cope and adapt. More interestingly, we only observed extinction of populations under certain treatment regimens, particularly under random changes and fast regular changes, already indicating that these 2 treatment types impose particular high levels of selection. We then looked at adaptation rates in the surviving populations, and they will be illustrated here in this graph. On the x-axis, you see time. On the y-axis, you see a proxy for adaptation rate. And then the different treatment types are indicated by different colors. These are the results. In the end, the bacteria always adapt. But initially, we see differences. And we see a slower rate of adaptation, especially for random changes of antibiotics and fast regular changes of antibiotics. So these results already highlight that exactly fast changes and random changes make it extremely difficult for bacteria to adapt, at least in the short run. We, of course, wanted to understand why such sequential treatment protocols make it difficult for bacteria. And we tested a variety of different alternative explanations. And to cut a long story short, we found out that the changes between antibiotics induce particular physiological changes in the bacteria. And it is these inducible physiological changes that may make it difficult for bacteria to cope with sequential protocols. So to explain that in more detail, we assessed physiological changes upon antibiotic switches with a specific treatment. And so now you should see my mouse as a red dot. So what we did, we took bacterial culture in log phase and then subjected them to a pre-treatment of only 15 minutes with either one antibiotic, another antibiotic, or no antibiotic as a control. Then we completely changed the media and then exposed these cultures to a second antibiotic, in this case, gentamicin. So what we try to do here, so we induce some kind of physiological change through the pre-treatment, the exposure to a first antibiotic, and then assess what consequences this has for bacterial survival in the presence of the second antibiotic. And this is how the results can look like. So here along the x-axis here again of time. Initially, this is the pre-treatment time, and then this is the main treatment time. And here on the y-axis, we plot a survival of the bacteria. In gray and dashed lines, we see the control. So there is no pre-treatment, and then we have the main treatment with gentamicin, and at some point the bacteria die. Most importantly, if we have a pre-treatment with either ciprofloxacin or carbonicillin, then we get enhanced killing by gentamicin. So demonstrating that this short exposure here induced some kind of physiological or cellular change that then makes the killing efficiency of gentamicin more efficient. So gentamicin kills with higher efficiency after this kind of pre-treatment. We did these pre-treatments in all possible combinations. So these are the drugs of the pre-treatment, these are the drugs of the main treatment. And whenever you see something in red, then we have enhanced killing in the presence of the second antibiotic. If you see something in blue, we actually have reduced killing. So red indicates higher killing efficacy. Blue indicates some kind of protective effect, some kind of cross-resistance, but not genetic cross-resistance, but physiological cross-resistance. And with this matrix, it already shows that all of these different possibilities are happening. We now took this matrix and then calculated a cumulative measure of this inducible effect across a sequential protocol of our main treatment, and then related that to either the adaptation rate or killing efficacy of the treatment. And we find a wonderful, nice correlation. So I will explain it in a minute. So here we actually have this cumulative measure of this inducible effect that we termed hysteresis. And the more we go to the right, the more we have sort of the negative inducible effect. And we then related that to extinction events and see that the more we have this inducible negative effect, the higher the extinction rates that we observed in our evolution experiment. And we found a negative correlation for multi-drug resistance and adaptation rate, meaning the more we have of this negative inducible effect, the lower the adaptation rate or the lower the level of evolved multi-drug resistance. So this very nicely demonstrates that antibiotic switches cause some kind of inducible physiological or cellular change that then increases extinction rates and lowers adaptation rates in sequential protocols. So the question now is this broadly applicable? And to test the applicability, we now characterize the occurrence of this negative inducible effect, negative hysteresis, in a variety of Pseudomonas strains with very different genomic backgrounds. So here you see genome phylogeny of all publicly available Pseudomonas aeruginosa genome sequences. And then the lines indicate the members of a panel that we established together with colleagues from the University of Hanover in Germany. And this is a panel that covers more or less the entire genomic diversity of Pseudomonas aeruginosa across the three main phylogroups, and also including both clinical isolates in purple and environmental isolates in green. So we characterize all of these different strains for the ability of responding to this inducible negative effect. So what you will see in a minute is then a matrix of the expression of this inducible negative effect. So here you have the different strains organized vertically. And then horizontally, you have an indication of different antibiotics used as a pre-treatment. And then in this case, gentamicin was used as the main treatment. Or alternatively, gentamicin was used as a pre-treatment and these other antibiotics as the main treatment. And then the same setup for ciprofloxacin. And whenever you will see something in red in a minute, then this is an indication of negative hysteresis. So the negative inducible effect. If you see something in blue, then it's positive hysteresis, so protective effect. And again, to keep a long story short, we see a lot of red in this area. And this is always a pre-treatment with a beta-lactam drug followed by an aminoglycoside. And that seems to lead to very robust expression of negative hysteresis across all of the genomic backgrounds. And this is, of course, extremely promising because it suggests that we can actually exploit this inducible negative effect across the entire species of Pseudomonas aeruginosa. It is robustly expressed across different genomic backgrounds. What makes it even more exciting is that this phenomenon is even expressed in strains that are resistant to the pre-treatment drug. And that is illustrated here. So here you see a lot of red. And this is for strains that are actually resistant to a beta-lactam pre-treatment drug, in this case, piperacillin tazobactam. So they are resistant, but if we now use piperacillin tazobactam as a pre-treatment drug, then this still induces physiological changes that then enhance killing efficiency in the presence of the second drug, the aminoglycoside gentamicin. So this points to even being able to target resistant bacteria, at least those that are resistant, say, to the pre-treatment drug. It did not really work if the bacteria were resistant to the main treatment drug, as illustrated here. So here we have strains resistant to gentamicin, and here we do not get a significant negative hysteresis result. The question is, how is this mediated? Is this sort of robustly mediated? And this is work in progress, but we have evidence that this inducible physiological effect is dependent on the CPX envelope stress response, and particularly dependent on a functional CPXS, that is a sensor that you find in the inner membrane, and that senses cellular stress or inner membrane stress. And this is also highlighted in these results here that I will try to keep short. So here we actually looked at intracellular concentration of the second drug of gentamicin, either in red with a pretreatment or without a pretreatment. So if we use a pretreatment, then we see increased intracellular concentration of the main treatment drug, gentamicin, and this is something we see across time. But if we now take a mutant of the CPXS gene, then this increased uptake of the second drug is no longer observable. So nicely highlighting that indeed a functional CPXS is required for the inducible negative effect, or the inducible increased killing effect. So with that, I'd like to switch gears and come to a second topic, and that is focused on collateral sensitivity. And the starting point was a similar experiment as I showed you before. So again, a long-term experiment where we had sequential treatment protocols, so where we switched between different kinds of antibiotics. In fact, we did this entire experiment 4 times. And we used different kinds of antibiotics, so either coming from 3 different classes, and that's what I've just shown you before. But we also had 2 control treatments where we took antibiotics from the same class with the same more or less mode of action, so all targeting the cell wall, all being grouped under the beta-lactam antibiotics. For example, in the case that I'm showing you in a minute, combining doripenem, carbonicillin, and cefzolidin. We included them as kind of controls because we knew that there should be some kind of cross-resistance between these antibiotics. So we expected that bacteria will be easily able to adapt to these treatments. Most surprisingly, they didn't necessarily do that. And this was best visible if we now looked at extinction rates in these evolving populations. And as a reminder, again, we always used sublethal concentrations of the antibiotics, so the bacteria should potentially always survive. But when we then looked at overall extinction frequencies for an entire evolution experiment, then this is what we show here along the vertical line. So the experiment that I showed you before showed an overall extinction rate of around 14 to 15%. But then surprisingly, we had this experiment 3 with a much higher overall extinction rate. And this is indeed the experiment where we combined doripenem, carbonicillin, and cefzolidin, so antibiotics of the same class. And if we now look at more detail, where extinction occurred, here on the y-axis and on the x-axis, you have different treatment types. Then we again observed extinction, particularly commonly when we had fast changes between antibiotics or random changes between antibiotics. So this really came as a big surprise. And now we try to understand what is causing this. And again, to cut a long story short, it has to do with spontaneous resistance rates towards different antibiotics and collateral sensitivity. So spontaneous resistance rate we established and characterized using the classical Luria-Delbruck fluctuation assays. And then we took these spontaneously resistant mutants and then subjected them to other antibiotics to look for the expression of either cross-resistance or collateral sensitivity. Collateral sensitivity is a higher susceptibility to a second antibiotic than observed before for the ancestor. And in this case, it's not inducible physiologically, but it is genetic because with a fluctuation assay, we select for genetically resistant mutants. And what we observed is that there is variation in spontaneous resistance rates towards the different antibiotics. The y-axis shows the resistance rate, and the x-axis shows the different antibiotics. And we see a significantly lower rate of spontaneous resistance for doripenem, for one of the antibiotics. Then we tested for collateral effects. And here you see the antibiotic towards which we had spontaneous resistance, so either doripenem, cefzolidin, or carbonicillin. Then we tested them for cross effects towards the same antibiotics. And whenever you see something in purple, then we actually have higher sensitivity towards this antibiotic. So for example, if there's spontaneous resistance to carbonicillin, we actually get higher susceptibility to doripenem than in the ancestor. So this is the information that we now took combined. In a particular sequential protocol, we can then consider to what extent we have the presence of antibiotics with low resistance rates. And on top of that, we can also consider to what extent we then have switches that lead to an increased sensitivity towards this second drug. So we calculated again a cumulative measure for resistance rates across a sequential protocol, and we also calculated a cumulative measure for these collateral effects across a sequential protocol, and then linked that to extinction rates. And these are the results that I'm showing you now. I start with the number of switches and its relation to extinction events, generally showing the more switches, the more extinction we get. But importantly, if we have sort of a high rate of a high rate of spontaneous resistance, as indicated along the x-axis here, then we get little extinction, meaning if the overall cumulative rate of spontaneous resistance is low, we get high extinction. And if this is for cross resistance, if we get a high level of cross resistance occurring at switches, we get low extinction. But if we actually get a low level of cross resistance, meaning a high level of collateral sensitivity, we get increased extinction. So this result shows that a sequential protocol can also be very effective if we have a sequential protocol that includes antibiotics against which bacteria have problems evolving resistance spontaneously. And if the sequential protocol also includes switches that often come with collateral sensitivity, so an increased level of sensitivity in case that resistance evolved to the first drug. So this can indeed increase the efficacy of a sequential protocol, even if it includes antibiotics from the same class. And I think this is the exciting finding. Usually antibiotics from the same class would not be considered for treatment by clinicians, because you would immediately think you can only make it worse. But our results highlight that it is possible, and this may actually point to new ways of improving antibiotic therapy. And I'd like to emphasize again the importance of collateral sensitivity. So the concept that I already used before, this is an illustration of how we can understand collateral sensitivity. So it refers to the phenomenon where the evolution of resistance to drug A automatically leads to an increase in susceptibility to a second drug. So this concept looks extremely exciting and promising, because even if bacteria are able to evolve resistance, they make it worse for themselves because they become more susceptible than before to a second antibiotic. So this is a phenomenon that could be exploited in sequential protocols, as just shown to you with the last example that I gave you. We've also shown, and I'm not presenting you the data, that this phenomenon can also be exploited to improve combination therapy. So where you combine two antibiotics at the same time. Because if you combine antibiotics that are related to each other with collateral sensitivity, then this makes it very difficult for bacteria to adapt. One important question is this a phenomenon that is robustly expressed by bacteria, and in this case, Pseudomonas aeruginosa? And that is the last data set or piece of information that I'm showing you today. So we first characterized the evolution of collateral sensitivity in Pseudomonas aeruginosa by subjecting bacteria to increasing concentrations of one drug and then checking for what kind of collateral effects may be possible. And these collateral effects that are possible are summarized again in a matrix, where along the y-axis, I show you the antibiotic to which the bacteria evolved high levels of resistance. For example, we had a couple of bacteria evolving high levels of resistance against ciprofloxacin. And these bacteria were then tested against the same antibiotics to see whether they then express cross-resistance, that is then indicated by a greenish color, or whether they express collateral sensitivity, which you will see in a minute in a purple color. The results are always shown by 10 bars, indicating the 10 independently evolved replicate populations, or in this case, Pseudomonas aeruginosa strain PA14. And this is the overall pattern that we get. So you see actually a lot of variation. What is also interesting is that we have particular combinations where All of the strains evolved high resistance against cefsolidin, but then some of them showed collateral sensitivity against gentamicin, but other populations showed cross resistance. And this effect was dependent on the exact evolutionary trajectory leading to resistance to cefsolidin, which we showed in our study and published previously. What is more important and exciting for us is that we also have cases where you just see the purple color. So evolution of resistance to ciprofloxacin seems to lead to very robust expression of collateral sensitivity towards the here included aminoglycoside drugs, gentamicin and streptomycin. So this looks extremely promising as a case of collateral sensitivity that may be exploitable for medical treatment. The question still is this robust? Or is this dependent on genomic background or the exact path of evolution of resistance? What makes these results even more promising is that by coincidence, a colleague of ours, Sara Hernando Amado from Madrid in Spain, she actually performed a series of studies where she found more or less exactly the same results. So whenever she evolved high levels of resistance against ciprofloxacin, she could also identify very robust increased sensitivity towards aminoglycoside drugs, for example, tropamycin. And I will not explain these results in details, but they actually more or less confirm these results that indeed we seem to have found a very robust case of collateral sensitivity. And this robust case strongly suggests that this is clinically exploitable. With this, I'm at the end of my talk and presentation. I argue, and I hope that I could convince you that evolutionary concepts can help us to improve antibiotic therapy, and in this case, sequential therapy. And it's different phenomena that may be responsible for that, including inducible physiological effects that we termed negative hysteresis, fast switches in general, but also the phenomenon of collateral sensitivity. Additionally, we could show that robust negative hysteresis is expressed in Pseudomonas aeruginosa upon switches from beta-lactam drugs to aminoglycoside drugs. And robust collateral sensitivity is observed in Pseudomonas aeruginosa after the evolution of resistance to ciprofloxacin and then leading to increased levels of susceptibility to aminoglycoside switches. And with this, I'd like to thank you for your attention.
Thank you very much for your details talk about how to apply evolution principle to optimize antimicrobial therapy. Yeah, so the next one is the science observed between cipro and aminoglycoside seems to be dependent on the two mutations. How should combination antibiotic therapy be designed to ensure that the resulting cipro-resistant will be limited to this mutation and not other genes?
Yeah. That's an easy answer because it is actually, the question is based on a misunderstanding. I showed you more detailed results for two genes that we knocked out to emphasize that we can more or less recapitulate the pattern of collateral sensitivity by creating these mutants in the lab. We have done that for other mutations, so mutations in other genes, and obtain the same result. So for us, it is really, really fascinating to see that irrespective of which genes are mutated to cause resistance against ciprofloxacin, all of these different mutated genes always cause collateral sensitivity to aminoglycosides. So far, we have not found a single case where this is not the case. So this is really exciting. So there may be perhaps coming up sooner or later, you never know. There may be a case where we actually cannot recover that. But so far, we always could observe it. And that makes it so convincing or so fascinating that this is really a very robust case of collateral sensitivity that should be exploitable for improving therapy. Okay.
Right.
The question is, have you tried this phenomenon to test this phenomenon in mice or other preclinical models? No. So we have initiated work in mice with collaborators, but it's apparently more difficult. And the problem is perhaps our focus on Pseudomonas aeruginosa, which is usually considered as an important pathogen of lungs. And the lung infection models in mice are not easy and also not really that similar to the human situation. Therefore, at the moment, we've also more or less decided to abandon these mouse experiments and rather focus on observational studies in a patient context. And this is something that we have initiated. And here we take advantage of the fact that such changes in antibiotics are actually part of standard care, at least in Germany, for other reasons, allowing us to do at least an observational study on such antibiotic switches. And I hope that in the not too far away future, we'll be able to present some results on this.
Yeah. So next one should be similar with the in vivo model, but the effect of the innate immunity.
Mhm. That's so the question by Nabil Hayek, to what extent we should rather use a combination of different antimicrobial peptides. That's a fantastic question, and this is something that colleagues of ours are actually pursuing. It's actually a couple of groups that are looking into that. Best known to me is a colleague based in Berlin. They're looking at whether we can combine antimicrobial peptides in an effective way to minimize resistance evolution. And the results that they obtain are actually very, very promising. Since others are doing that already, we are not doing that. But I find this an important line of research, again, with the aim of using evolutionary knowledge, evolutionary information to improve therapy.
Thanks. We have question from Will Smith. Do you see any evidence that collateral sensitivity across resistant phenomena are environmentally dependent?
That's also a very good question. We have not specifically addressed that because for us, we first found it important to actually understand whether the exact evolutionary trajectory leading to resistance has an influence. What I can say so far is that these patterns of collateral sensitivity, at least in relation to ciprofloxacin and aminoglycoside drugs, have been quite robustly observed for Pseudomonas aeruginosa in different labs using slightly different approaches and media conditions. So simply taking that into consideration, I'm at least slightly optimistic that it should also be robust to different environmental conditions. But so far, I cannot entirely prove it. But it's a very good point.
Thanks.
The next question is, what is more effective, sequential therapy or combination therapy where you simultaneously combine different drugs? This again is a very exciting and interesting question. We have done a very large study a couple of years ago on the efficacy of combination therapy. We have not directly compared it to sequential therapies because it's not as trivial as it sounds. Because to implement the relevant controls is not that easy. At the moment, I would say both are actually valid alternatives that should be pursued. And I'm quite convinced that both strategies should help us to improve therapy. So combining antibiotics simultaneously, but also using them in sequential protocols. What is perhaps important is that we can provide ideas which kind of antibiotics should be combined, taking into consideration the here presented evolutionary concepts, such as collateral sensitivity. Then the question is whether negative hysteresis also occurs in other bacteria. Initial data by colleagues suggest yes. So there is some initial work on E. coli that strongly suggests that it occurs also in E. coli. Colleagues of ours have also looked into that in even Mycobacterium tuberculosis, where this is much more challenging because of the much longer generation time, and they could also confirm it. So it seems to be a phenomenon that is applicable across different bacteria, but much more research is currently needed to better understand it. So then there's another question on applying the antibiotics in a more dynamic way, because that is much more relevant for the clinical or patient context. And that's, of course, a very relevant point. We have not done that yet. We have done some experiments where we varied antibiotic concentration in our experiments. And this very clearly has an influence. And I fully agree that this is something important to address in future work. Yeah, and this is a question whether porins or efflux mechanisms are blocked. I can imagine that this is a question relating to the mechanism underlying collateral sensitivity. And it is also something that we are currently looking into. We're trying to understand how exactly or what exactly is causing collateral sensitivity. We have some, or let's put it that way, these processes are not completely independent of each other. There is a strong indication that efflux is important, but it can be important in two ways. So either it could be that efflux is blocked, or that a change in the activity of efflux pumps leads to a change in proton motive force across the membranes that could then enhance the uptake of aminoglycoside drugs. And these are two alternatives that we are currently looking into. Yeah, Suni Wu asks to what extent the sequential protocols that we are considering can be actually applied in a patient context. And this, of course, depends on guidelines for antibiotic therapy in different countries. And I'm most familiar with the guidelines in Germany. And here, such sequential protocols are possible. It depends on the type of infection, of course, and the type of pathogen. For Pseudomonas aeruginosa, there is a suggestion that if aminoglycosides shall be applied, that these should only be applied for a very short time period and then should be alternated with, for example, a beta-lactam drug. And the reason for this setup is it has the aim to reduce exposure to aminoglycosides that can have very strong side effects. But it is interesting for me that such switches are actually already in place, but for completely different reasons. And this is what we are also currently looking into together with our collaborators from the clinical field within the framework of an observational study, whether indeed this kind of setup also leads to lower resistance, lower rates of resistance evolution because of the phenomena of negative hysteresis that I illustrated in my talk.
Yeah, I
Okay, I
probably
think
have
these were all the
another
questions.
very quick question. So could you please, in terms of the lot of like already existed large data sets from your group, can you please comment on is it possible to apply AI or neutral large language model like to integrate those evolutionary and genomics data, which probably can used to predict the evolutionary path under different selective pressure, or like the mutational hotspot are likely beneficial on compensatory mutations?
Again, a very good question. I think I have to answer in a careful way. Of course, AI approaches are now used more and more to also help improve therapy. What is important is you need good training sets, and the training data set already needs to be quite comprehensive and data rich. And I think that we are currently still lacking good training sets. But this is exactly something that should be accomplished in the next years to have better training sets, because then we can actually train an AI algorithm to improve predictions also of medical relevance. So I think it's a good point, but I think one should always keep in mind that AI approaches will not necessarily provide all of the answers in the world.
We actually have another new question. How stable is cholesterol sensitivity over time in bacterial populations?
That is also an extremely good question. So, can bacteria or are bacteria able to counter collateral sensitivity? And this is something we also looked at in one study that we published a few years ago. We have to face it, bacteria are masters of evolution. So in the end, they will always win and always manage. So the outcome of our study was that bacteria are able to counter collateral sensitivity, but there is a variation in how fast and how well they are able to counter this. And this is perhaps the little hope that we have. So it seemed that if we still apply the first drug against which resistance evolved initially, so if we still include that when we then also administer the second drug, then we make it more difficult for the bacteria to counter collateral sensitivity. So this is one conclusion that came out of that study. And another conclusion is that it may also depend on the exact resistance mechanism and or also the exact drug towards which collateral sensitivity is shown. So it seems somewhat more difficult to counter collateral sensitivity that is expressed against aminoglycoside drugs.
Sure, thanks. I think we are about the time. So if there's no more question, please join me to thank again for Professor Schulenburg's time and the exciting talk.
Yeah, that was a pleasure for me. Thanks a lot
Thank you very
and
much.
take care.